Good morning, my microbiology students. Welcome to class. And today we're going to be covering chapter 5 of our text, which covers viruses and prions. So as I've stated before, we're not going to cover the entire chapter, just the things that are relevant to our state standards. So in this chapter, we're going to cover 5.1, 2, and 3, but not the latter half. So in 5.1, we'll talk about what viruses are and how they relate to the biological spectrum. We'll talk about the general structure of viruses, and then we'll talk about their reproductive cycles. So first and foremost, our learning outcomes for the first section is to explain what it is meant by when we say viruses are filterable, and to identify better terms for viruses than alive or dead. So to begin with, viruses are capable of infecting other cells, and they're capable of infecting every cell type. This includes obviously animal cells, because we know that we can contract viral infections, but also they can infect other cells. They can infect plant cells. Plants can get viral infections. So can protists. Fungi, algae, and even bacteria are capable of being infected by a virus. And we've mentioned previously how ubiquitous bacteria are, meaning they're everywhere. Viruses are even more prevalent, up to 10 times more prevalent than bacteria are. Seawater, just to give you kind of an example of how prevalent viruses are in the environment, they can contain 10 million viruses per milliliter. And that's a relatively small amount of fluid. So when did we discover bacteria? They were discovered with very primitive microscopes around 1678. That's about 345-ish years ago. And they were recognized to cause disease later than that in 1876, which is approximately 145 years ago. We knew, however, that some diseases were caused by something else. Some examples: polio, smallpox, but we just didn't know what it was at that time that was causing these diseases. We knew bacteria caused disease, but we also knew there must be something else to cause disease too. Now, how did we know that? Well, in around the 1880 to 1890s, we determined that tobacco disease was caused by something smaller than bacteria. Foot and mouth disease in cattle was caused by something smaller than bacteria. And again, how did we know this? Researchers determined that when we have infectious fluids and we take it from a host organism, an organism that's sick, and we pass it through a filter. There are porcelain filters, and those porcelain filters were designated, that was their job, to trap bacteria. Then the filtrate still remained infectious. The filtrate is the liquid that's left over. So to give you kind of an illustration of this, I have a colander here, strainer. And so if you're trying to filter your lettuce from the water that you're cleaning it with, the water will filter out. That would be the filtrate. Filtrate, what leaves the colander, and what's trapped inside it in this case is the lettuce. Here we have a HEPA filter. So as y'all know, airflow will travel through the filter and it will trap large or even small debris depending on the filter. These HEPA filters will trap small debris like even viruses in the filter itself, and then what will pass through it is clean air. That clean air would be considered the filtrate. Sorry about that. I can't finish my word there, but it was supposed to say filtrate. So anyway, they did the same kind of thing with these infectious fluids. They put an infectious fluid through a filter, and so theoretically any bacteria would be trapped in the filter, but the filtrate was still infectious. So that told them there must be something smaller that can cause disease besides just bacteria, and thus the term viruses was coined. It's a Latin-derived word that means poisonous secretion. Alright, so that's how we determined that something smaller than bacteria causes disease, and that was called a virus. So this is showing you an example of the experimentation the researchers did to determine this. So what you're seeing in this first image is a scanning electron microscope image. It's showing in the background, that would be the filter. That filter was located here, and it has little pores, these holes. And these holes were relatively, pretty small. We're going to say about 5 micrometers. And what we're seeing here, these rods throughout, these are bacteria. And as you can see, they are being trapped by the filter. They're too big to pass through those small pores. So, if we look at B here, the size of the pores on that filter is going to determine then what's captured and what's not. All right, so here what we're showing is the red balls are animal cells. The bacteria, so I'll point to it right here, animal cells, which is right A. The blue balls would be, as it states, bacteria, B. And then the viruses are the green balls, and I'll write V for virus. So with this pore size on this filter of 5 micrometers, we will capture animal cells. However, our bacteria cells in blue, as well as the viruses in green, are able to pass through. So if we decrease this filter pore size to 0.2 micrometers, very much smaller here, we have 5 micrometers compared to 0.2 micrometers, that is now going to prevent bacteria from passing through. So bacteria do not pass through. However, and of course also let me point out, animal cells don't pass through either because they're bigger than bacteria. So that pore size only allows for things that are smaller than bacteria to pass through, such as our viruses. And so then what they observed is that this filtrate, that's again the filtrate, the fluid that passed through, contained infectious particles. So there was something smaller than bacteria that was capable of causing disease, and they called them viruses. Alright, so let's look at the virus sizes, how big they are. So until the 1930s, when the electron microscope was invented, no one had seen a virus because they were so small. The current microscopes at that time were not powerful enough to do so. Viruses range from about 20 nanometers to 900 nanometers in size. And so what this picture is showing below is some various viruses as well as bacteria and a eukaryotic cell, so you can kind of see a comparison of sizes. So the largest cell here is a budding yeast cell. If we look at the smaller one here, that's a bacteria, E. coli. It's a rod shaped. We have a coccus here, which is round. It's only 1 micrometer in diameter. And then we see several viruses. So here we have one of the larger viruses, and they get smaller and smaller and smaller and smaller until you get all the way down to the bottom here. And in fact, here's something of interest just to give you an idea on how small this is, but 50 million polioviruses, that's this one here, could fit into the average human cell for reference. So very, very tiny. So what we know now about viruses, and this relates to one of our learning outcomes about describing how they fit into this biological spectrum, is that they are not cells. We consider them acellular. They are not cells. That's why we would consider them not to be a living thing. Again, one of our learning outcomes is to explain why that viruses can be better classified than alive or dead. Because they're not a living thing, because living things are required to be made up of cells. That's one of the characteristics of living things, is they must be made up of cells. But again, viruses are not. So what we're seeing here, viruses are not cells. They are not cells. Instead, they are obligatory intercellular parasites. And what that means is that they are required to parasitize cells for their survival, quote unquote, survival again in quotes because they're technically not a living thing. They cannot reproduce independently. They cannot make enzymes for most metabolic processes that living things need to be able to do. They don't have machinery for synthesizing proteins like ribosomes, etc. So what they do is they take control of the host cell that they infect, and they use its stuff to do those things. They use its organelles, its ribosomes, its enzymes to help manufacture more viral particles that can ultimately be released to spread the virus. So they are not cells, they are not a living thing. So there are two sides of the debate, though, ultimately. Since viruses are unable to do those various things that we listed independently, many scientists say that, well, they're not living things and should be called infectious molecules. Even though viruses do not exhibit most of the life processes of cells, they can direct them and thus are certainly more inert than lifeless molecules. So there is some arguments among science as whether, or not they're technically alive or dead. But most textbooks and the way most scientists will describe viruses at this time is that they're either active or inactive rather than alive or dead. So our next topic of conversation is going to be viral architecture. We're going to describe viral structure. What we're seeing here in the pictures are various images of viruses, and you can see their structure can vary. Here we see what we call a negative stain. It's of an influenza or several influenza viruses. We see Ebola stains. This is called a positive stain. And we see the Vaccinia virus. So just different ways to image them. But all of these viruses and all other viruses, all viruses in general, contain only the parts that are necessary to hijack and control a cell. And that includes two things: a capsid and a core. Alright, so all viruses contain a capsid and a core. The capsid is a repetitive pattern of proteins that creates a coat. So that covering is kind of, I guess you could say, it's analogous to the plasma membrane for cells, which is made up of phospholipids. But in this case, this covering, the boundary that separates the inside of the virus from the outside, is made up of proteins, and it's called a capsid. Individually, the protein units are called capsomeres, and collectively, it's called the capsid. Some viruses, however, also have an envelope that surrounds that capsid, but not all of them have that. The core of the virus is always going to have some kind of genetic material, which is in the form of DNA or RNA. And some viruses will have additional enzymes and matrix proteins, but not all. So all proteins have a capsid made up of protein. They have a core that has genetic material, DNA or RNA, and then some have these other things like envelopes, enzymes, and matrix proteins. So another term you should be familiar with is naked viruses and nucleocapsids. Naked viruses only consist of a nucleocapsid. If we break that down, nucleocapsid, it contains a capsid, and nucleo is referring to the genetic material inside, because genetic material, whether it's DNA or RNA, are called nucleic acids. So this is the genetic material and the capsid, which is the protein coating. Some viruses, remember we said on the other hand, have an envelope. Well, for those viruses, they still contain the nucleocapsid, but they're not considered naked, all right, because they contain an envelope around it. So they have the capsid, which is shown in the, I guess that's kind of like a light bluish color. And then the capsid, or excuse me, the nucleotides, there we go, are located inside, but additionally it has this envelope that's surrounding it all around. So this is called an enveloped virus. So it contains this additional covering that's typically going to be a modified piece of the host cell's membrane. Now, you can also find spikes on viruses, either naked or enveloped. So you'll see them, I'll point to one here. And also, of course, we see some various spikes on this one here. These are important. They project from the nucleocapsid or the envelope and allows viruses to dock themselves to other cells, like host cells. They have to do this in order to infect a cell. So all in all, again, our naked viruses contain only a nucleotide and a capsid, the nucleocapsid. Envelope viruses contain both of those things, but also an envelope around it. And spikes are found on both that assist in docking of the virus to a host cell. So let's talk some more about that capsid. So in the previous pictures, they've pretty much all been an icosahedral shape. I kind of relate that to like these fancy multi-sided dies, or dice, plural. But the capsid is the most prominent feature of the virus. It's something that's the most visible. It's constructed from these identical protein subunits that we said we call capsomeres. And they can form three-dimensionally in three different types. So the first one is icosahedral. So you can imagine these dice as viruses, if you will, where each die is going to be made up of small subunits of proteins called capsomeres. And collectively, they form a three-dimensional shape that is icosahedral. So next, we have helical shapes. So here we see the genetic material in red, and then these yellow little, what's pictured as yellow little balls, these would be the capsomeres, those proteins that make up the capsid. And then finally, we have the complex shape. This is where it doesn't fit into the other two, so it has different shapes other than helical and icosahedral. Now, for our enveloped viruses, we said that they have a portion of our host cell's membrane. They take this when they are released from a host cell. So what we're seeing is this virus here, and it was infecting, or I guess it's still technically infecting the cell, it's still in it, but you can see it's pinching off. It's trying to escape the host cell to go on to infect another cell. And so when it does that, it's taking a little piece of this plasma membrane of the host cell that will then encapsulate the virus that is leaving, this enveloped virus. Alright, so next, at the core of a virus, we said that's where the nucleic acid is, the viral genome. It can be made up of DNA or RNA. So that's different than cells like animal and plant cells and bacteria and so on. Previously, when we've talked about genetic material, it's been DNA that's found in the cell that is our genetic material. But viruses, they don't follow those rules. So they can be DNA viruses or they can be RNA viruses, but they're never both. And structurally, they're a lot more complex than what we have learned in the past as far as viruses go, and as far as you may have seen in Bio 1 with me, or if maybe you might have done a little bit of this in AMP. But ultimately, we're not going to go into a lot of details here, but DNA can be double or single-stranded in viruses, and that's weird because what you should have learned in the past is that DNA is double-stranded. Well, it usually is. 99% of the time, when we talk about any other cell type, really, it's double-stranded. But sometimes in viruses, it's not. So it's weird. RNA, we've historically learned, or you've learned a prerequisite, is single-stranded. However, there is double-stranded RNA that's possible in viruses. There's also another classification that we could call viral RNA, positive sense or negative sense. I actually put an equal sign, that should have been a positive. We're not going to go into the details on what that means, but my point is to show that it's really complex compared to what you have learned possibly in the past. It can also be circular, linear, segmented, which is very strange because we've only learned about, everyone probably has learned about linear DNA. If you took Bio 1 with me, we talked about circular DNA and bacteria, but viruses can do some weird stuff. So, next, the number of viral genes is quite small compared to that of a cell. For example, in the hepatitis B virus, there's only 4 genes. Now, there's hundreds in some of the herpes viruses. Humans, on the other hand, as a non-viral entity, we are an animal, we have 23,000 genes. And so again, that's because these viruses are only going to possess the genes they need to invade a host cell and interrupt its activities to do what it wants it to do. Alright, so next we're going to now describe how animal viruses multiply. And listen up, because it gets a little crazy in this next section. I feel like this is probably one of the harder concepts in micro from what I have experienced so far teaching it the past several years. So get your coffee. Viruses are tiny parasites, again, that seize control of a cell and its machinery to make more viruses. And what we're going to describe first is how animal viruses multiply. So we're talking about animal viruses right now. Remember, there's other types. We have bacterial viruses, plant viruses. We're focusing on animals. The general phases of the animal viral cycle is as follows: absorption, penetration, uncoating, synthesis, assembly, and release. So what I've done over here to the right is I've given you a mnemonic device: acrobatic, public, unicorn, C, athletic, results. And here's my acrobatic unicorn. So you can use whatever mnemonic you want. This is just something I came up with. But what it's telling me is each one of these letters at the beginning is representing one of these steps to the left in order. So it gives you a way to help you remember something else. So the A here in acrobatic will stand for the A in absorption, public, penetration, unicorn, uncoating, and so on and so forth. And maybe this little silly picture will help you remember it too. Alright, so let's go through each step. The first step is, again, absorption. This is where the cell is unlocked, quote unquote. This is where a virus will dock itself to a compatible protein on the cell surface and bind to that receptor site. So hopefully, I know you learned this with me in Bio 1. I'm sure y'all learned about how enzymes and proteins work in an AMP as well, but just in case. So the way that proteins work is they're going to have a very specific shape, a very specific three-dimensional shape. And so for a moment, let's pretend here that this is a protein on my plasma membrane, and here's my plasma membrane. I'm not going to draw in all the phospholipids because we'd be here all day, but this is the plasma membrane and a protein that's embedded into it. Notice it has a square shape here. So whatever fits this protein receptor, whatever's supposed to bind to it, will have a complementary shape. So let's say this is some sort of signaling molecule. I'm going to write SM for short. It will bind to that protein receptor site and then result in some kind of response. The cell will do something in response to that. Now, what viruses do is they mimic these receptor protein sites in such a way that they fit into them. Alright, so what we're seeing here is a virus that is going to have these spikes that are going to have a certain shape that will fit some of these protein receptors. So, and again, we need to make sure that we reiterate some information that is relevant to this. Not all cells have the same pattern of receptor proteins. For example, your red blood cells are going to have a different pattern of proteins embedded in the plasma membrane compared to your immune system cells. So that restricts the host range. You know, our hemoglobin, or let me backtrack there, our proteins that are embedded into our various tissues as humans or as animals are going to be different than those that are in plants and other groups of organisms. And so why that's relevant here is because it restricts what we call host range. So not all viruses can infect anything. they infect specific cells that have certain protein receptors. So, for example, hepatitis B, it infects the liver cells of humans. It doesn't infect our brain cells. It doesn't infect our immune system cells or our muscle cells. It's specific to liver cells because liver cells have a receptor site that other cell types don't, that this virus can interact with. The poliovirus, it infects intestinal and nerve cells of primates, so it's not limited to humans in this case. It can infect any primate, and it infects specifically their intestinal or nerve cells. And again, that's because those cell types of those specific groups of organisms have the certain protein receptors that they are capable of interacting with. So the poliovirus isn't going to infect a fish because it's not a primate. Its receptor sites for that the poliovirus would interact with are not the right shape for it to do so. Rabies, they can infect various cell types within the body of mammals only. So we would not see the rabies virus infect a plant. It would specifically have to be an animal in the group Mammalia, mammals. So that is restriction of host range. Alright, so that is how our viruses are going to adhere and absorb into the cell. So next we have penetrating and encoding. So I put these on the same slide. So we have the flexible cell membrane of the host is going to be penetrated by the virus, and it's going to be taken in by either two processes, one of the other, either by endocytosis or direct fusion. So what we're seeing in this image here, we'll go with the left side first. We are seeing specific attachment, and again it's specific because it has to have the right protein receptors for the virus to dock onto. And then we're seeing this invagination here of the plasma membrane that is engulfing around this virus. And that engulfment creates a vesicle around it. That's what that is. It's the remnants of the host cell's plasma membrane that's now encircling it. So that is endocytosis. And if it helps you to remember, endo means inside and cyto means cell. So endocytosis is the engulfment of something into a cell. So you just need to, remember that it is going to cause this virus to be encapsulated by the host cell's plasma membrane, as seen in the yellow. Comparatively, if we look at this side, this is showing you direct fusion. So if we have a virus that is infused directly into the host cell, we'll see specific attachment again. We see membrane fusion, and then we see the entry of a nucleocapsid. All right, so compare here to here and see how it's different. In this scenario, when we have the plasma membrane invaginate and then surround the virus in a vesicle, that's this scenario here, scenario A. In scenario B, we have the virus that is going to adhere to the plasma membrane, but it doesn't engulf it. It simply fuses into it and passes. So this is the entry here of the nucleocapsid. You can see it has now been dumped into the cell. It's not surrounded by a vesicle like it was in scenario A. So then in step 4, we're going to have the encoding of the nucleic acid. So our capsid breaks down and it frees the DNA or RNA, so apparently, as well it could have been, of this virus, so it can now move on to the next step. Likewise in scenario B, the virus is in a vesicle in this scenario, but that envelope and the capsid break down, which uncoats the nucleic acid, which is now free to do the next step. So again, it just depends on the virus type on which pathway they'll take. If it's a naked virus, it will do direct fusion because it doesn't form an envelope. If it is an envelope virus, it'll utilize scenario A. But the ultimate goal here is to get the genetic material of the virus inside that host cell. And again, that's the DNA. Or again, could be RNA because it could be an RNA virus. But in this particular picture in the book, they use DNA. So our step 4 is synthesis. This is replication and protein production. So if we think of synthesis, we should be thinking of building, because when we synthesize something, that's what we're doing. We're making it. If you synthesize a cake, you take the ingredients and you bake the cake. So if we have a DNA virus, it will enter the host cell's nucleus and will be replicated and assembled there. And if you recall from either AMP or Biol 1, the DNA is located in the nucleus. So that's why our DNA of our virus is going to travel to the nucleus. That's where RNA is made. So just a brief reminder, how do we make proteins? DNA is converted to RNA, which is converted into protein. These processes were called transcription and translation, right? Transcription is transcribing DNA to RNA. Translation is translating RNA to protein. So again, if we have a DNA virus, we're starting with DNA, it needs to be transcribed to RNA and then translated to protein. So that's why our DNA will travel to the nucleus of the host cell where that process is performed, transcription. On the other hand, if we have an RNA virus, it doesn't need to be transcribed because it's already in RNA form. So it will instead go through the process of translation by utilizing the host cell's machinery, like its ribosomes, in order to make more viral protein and then assemble it into the correct structure. And so that's going to happen in the cytoplasm because that's where translation happens in host cells. Now, there are some examples of some weird things that viruses can do that are kind of cool, but retroviruses, for example, they can turn their RNA genomes into DNA, and then I guess it's called that because you think retro, it's like going backwards. So instead of going straight to RNA to protein, they'll convert their their RNA back to DNA before they go on to do other processes. And it's kind of neat. So number 5 and 6 is assembly and release. So now that they've made multiple more viral particles, they are going to assemble them. They get put together using all their parts they manufactured in the previous process of synthesis. And now their goal is to release these new virus particles called virions into the environment near the cell. So what we're seeing in this picture, we have our capsid and our RNA. We see it assembling into a new viral entity, and then it is going to exit the host cell, in this case by budding. And so now we have a new free infectious virion with an envelope ready to infect another cell. So in reality, we're not just going to have one replicated virus exit the cell. There's going to be tons. So for example, if this was the pox virus that can cause a smallpox infection, for example, we would have 3,000 or 4,000 of these virions exiting this cell. If it was the polio virus, we're talking hundreds of thousands. So there's an immense potential then for very rapid viral proliferation, meaning viruses infect and spread in the body very quickly. All right, and how does that affect the cell? So we call the effects of these viral infections CPEs, or cytopathic effects. Cyto meaning cell, path meaning a disease state. So like pathology is the study of disease. So the cell disease effects. What is it? What happens to the cell when it's infected? But it causes damage to the cell, and there's different damages that can occur. Sometimes cells can have a very influential impact in their shape and size. Excuse me. They can have intracellular changes, which means they can have changes that occur in between the cells that make interactions occur differently. They can have inclusion bodies that can start to form, and even syncytia. I always have a hard time pronouncing that one. Syncytia, which is the fusion of multiple damaged host cells into a single large clump of multiples of cells that have multiple nuclei, we also call them giant cells. But you can see some of these cytopathic effects here in some examples. So here we have, normal, no virus. With polio, the cells are killed completely. They shrink and detach away from the surface, and they lyse, which means they rupture. So you see no cells. Here we have an adenovirus. These are going to cause intracellular changes that is going to affect the way these cells are organized. So you can see they're all kind of clumped together. Our RSV virus, we'll see adjacent cells will fuse together. Now we have giant cells with multiple nuclei. So that's where that term comes from with respiratory syncytial virus. It comes from that word that causes that means the fusion of multiple cells into one giant cell. And then rabies virus, we can see the formation of inclusion bodies throughout. Okay, so moving forward, some viruses are persistent. Comparatively, some viruses are not. So with certain viruses, let's say the stomach bug is typically caused by a virus, and it'll pass, and it won't come back unless you come in contact with it again. On the other hand, some viruses can remain what we call latent. They remain latent in the cytoplasm, and they can stay there for weeks, months, years, or even a lifetime. They're basically just kind of hanging out, is what I like to describe it as. They're not causing, I don't know how I did that. They're not actively causing an infection, but they're still there. Some viruses can incorporate their DNA into host cells to form what we call proviruses. Know that term. For example, the measles virus, if you can track the measles, it hides, quote unquote, it goes latent in the brain. But it can cause progressive damage over time. So these viruses that we consider latent, they can periodically become activated depending on certain stimuli. Some of them are like stress, if you have some kind of immunosuppression, an illness, pregnancy, etc., that can cause that virus to reactivate and emerge out of the latent state and be active again. And a great example of that is herpes simplex virus. So this is the virus that causes cold sores as well as genital warts. And another example is the herpes zoster virus, which is the one that causes chickenpox. So these, if you think about cold sores, some people will get a cold sore and it'll go away. What causes the cold sore is the herpes virus being activated. Why is it being activated? Due to some sort of stimuli. But then the cold sore will heal and they'll have a period of latency. And then it can be reactivated. Same thing with chickenpox. If it's reactivated, it will result in shingles, however, in this case. But it's caused by the same virus. It's just a reactivation of it later in life, usually. Additionally, some viruses are associated with cancer. About 13% of cancers are caused by viruses, in fact. So how is transformation? So viruses carry genes or produce proteins that can induce abnormal cellular function. So if we have a cell that has been transformed virally, it can cause that cell to increase the rate of growth, how fast it multiplies. It can change their chromosomes. It can change the surface molecules that are on a cell's plasma membrane, like the proteins it contains. and it can actually change its capacity to divide to the point that it's indefinite, which is essentially what cancer is. It's this uncontrolled cell growth. Now, I do want to take note here just a moment. Just because you contract a virus, any of these viruses that I'm listing here, it does not mean that you will get cancer. It's just a risk factor. And if you took biology with me, then you'll know that cancer is really complex, and that I related it to bubble wrap in class, how you have lots of, a big sheet of bubble wrap, and all these bubbles on the wrap can pop if you have this risk factor. So this would just be one bubble on the bubble wrap, if you can kind of help imagine it that way. For example, if skin cancer, if I go to tanning beds, I might pop a few of those bubbles. If I have a genetic predisposition, I could pop a few more bubbles. So it's not like just because you have one of these viruses that means you're going to get it. Okay, so I just want to clarify that so I don't have any panic in the class. But some examples include the papilloma viruses. These cause cervical cancers. We have the herpes viruses. They are associated with Burkitt's lymphoma, especially Epstein-Barr virus, which causes mononucleosis. We have the Hep B virus, which is associated with a risk factor for liver cancer. And our HTLV1 virus, which is associated with adult T-cell leukemia and lymphoma. So now, moving forward, we're going to talk about viral multiplication again, but notice we're going to talk about bacterial viruses. So these are viruses that infect bacteria. They are called bacteriophages, or I've heard people pronounce it bacteriophage, tomato, I say bacteriophage, but that is a virus that infects bacteria. Their structure is they are going to have an icosahedral capsid, called the head here. See multiple triangle shapes that form a three-dimensional shape. A central tube, as seen here, and these tail pins, as seen here. They look very alien-like to me, almost like a little spacecraft. That's the tail pins. So these tail pins, what they do is they will land, which is kind of funny because, like I said, it looks like a spacecraft. It lands on the bacteria cell, and the pins will connect to the bacteria cell, and they'll contract, and that will cause this bacteriophage to essentially inject its genetic material into the cell. So it's almost like a needle, like it injects it into the cell. So here we see our viral nucleic acid, whether it be DNA or RNA. So now, then what happens next? So there are two different pathways that our bacteriophages can take. So if we start with number 1 in the book, this is showing you the bacteriophage has injected its injected its viral genome into the bacteria. So it has been absorbed and it has penetrated, going on to number 2. And then there's basically two different things that can happen. That viral DNA can do one of two things. If it goes into the lysogenic state, then that viral DNA will infiltrate the bacterial cell's genome. So what we're seeing here, backing up, we have our bacteriophage. It's injecting its I keep on saying bacterial, viral DNA into the cell. It hasn't done so yet. It's about to. This here is the bacterial genome. Remember, if you took me for biology, you know that bacteria have a single circular loop of DNA. And if you were an A&P student, you didn't learn that, now you know. Bacterial DNA. So the bacteriophage is injecting its viral DNA into this bacterial cell. And if it enters into the lysogenic state, the viral DNA becomes latent as a prophage. All right, so this is called a prophage because we have this blue section, that's the viral DNA, that has been inserted into the genome of the bacteria. All right, so that's what a prophage is. Know that. It is the DNA that consists of our bacterial genome with the viral genome inserted into it. So to see that a little bit bigger, basically the stuff in blue is the viral genome and the bacterial genome is that in the pink. All right, so now that is the lysogenic state. And it might help you maybe to remember the word. It has gen right here, so think genetics. The genetics are combining in this stage if it's in the lysogenic state. So alternatively, we can have penetration occur and duplication of the phage, the bacteriophages components, and replication similarly to how we saw in animal cells. We see assembly, maturation, and then towards the end we will have lysis of the weakened cell because it gets so packed with all these bacteriophages it just cannot maintain, it cannot keep going. The bacteria cell ruptures and that releases these new viral particles that can then infect other cells. So it's important to know that's the lytic cycle when it continues on like that, but they're connected. We can have a virus that will infiltrate this bacteria, infiltrate it with its genome, and then go into the lysogenic state. Notice this arrow goes both ways. It can go into the lysogenic stage and incorporate its viral genome into the genome of the bacterial host, and then if it's activated to do so, it can splice out. So this viral genome can splice or split away from the bacterial genome and then go into the lytic cycle to cause infection and ultimately rupture the host cell to release more viruses. All right, so moving forward, what's the role in lysogeny in humans? So bacteriophages infect bacteria. An infected bacteria, though, can infect humans. So that's kind of a very interesting relationship there. Viral genes that are in the bacterial chromosome can cause some pretty nasty CPEs, those cytopathic effects. It can cause toxin production, for example. So lysogenic conversion are these traits that are acquired by bacteria due to bacterial infection by a virus. So it's giving these these bacteria new traits that can then cause us as humans more problems. So if we look at some examples, we have three bacteria listed here. We have Corynebacterium diphtheriae. This causes diphtheria, and it produces a toxin called the diphtheria toxin, and the reason why it's able to produce that is because of this lysogenic conversion, where there was a Corynebacterium diphtheriae bacteria that was infected with a virus that gave it that trait to be able to produce a toxin. Another example is Vibrio cholera. It produces the cholera toxin by the same mechanism. And finally, we have Clostridium botulinum, which causes botulism, and it is going to be able to produce the botulinum toxin. So I kind of like this little meme here. It's like here we have our initial bacteria prior to being infected with a virus, and now we've kind of got like a super bacteria here because it has viral genes that allow it to make toxins. All right. So lastly, I think that's the end. Yep, I have a video. I noticed last time that the video just sounded super weird whenever I replayed it, so I don't know if I'm going to be able to fix that, but I'm going to play this video anyway, or you can go into the PowerPoint and watch it if it sounds funny.
Chances are, in your life, you've had the common cold before. It can cause sneezing and coughing and can make you feel awful. The worst thing about the common cold is that you can take medications to help with the symptoms, but you really can't treat the actual cold with antibiotics like you would a strep throat infection or an infected tooth. Why? Well, let's talk about what causes it, a virus. The common cold is caused by a virus. While all living things are made of one or more cells, see the modern cell theory, viruses are not cells. Viruses aren't technically classified as living things since they lack many characteristics of life. But there is some debate in calling them non-living as well. Medications like antibiotics attack bacteria like E. coli, which are living cells. Antifungal medications can be used on fungi like athlete's foot, which are made of living cells. Viruses are not bacteria or fungi. You can't classify a virus as a prokaryote or a eukaryote either because it's not a cell. So what is their structure like?
First of all, viruses are much smaller than cells.
You typically are going to need an electron microscope in order to see a virus. Viruses come in many shapes. Our favorite one to draw is the bacteriophage. It sounds very fancy, but it's a type of virus that tends to attack bacteria. Despite this being our favorite to draw, know that the shape of viruses vary greatly. One thing all viruses have in common is that they have some type of genetic material. This genetic material can be in the form of DNA or RNA. Viruses usually have some kind of protein coat, also known as a capsid. The capsid can protect the DNA or RNA genetic material. Viruses can also have other structures. Some viruses package special enzymes with them. Some viruses have an outer envelope. These additions can be useful to the virus in virus replication. One thing that makes virus replication or reproduction unique is that they can't replicate without a host. And it's their reproducing that tends to be a problem for its host, whether the host is you, in the case of the common cold, or an insect, plant, this bacterium. Viruses tend to be very specific for the host that they're going to infect, but anything that is alive can be a host because cells have the machinery that viruses need in order to replicate. We're going to talk about two types of viral replication cycles. The first one is the lytic cycle. In the lytic cycle, the virus, remember very selective, attaches to a host cell. It often binds to a receptor that the cell has, which gives it access to attach. The virus can inject either its DNA or RNA, depending on what kind of genetic material it has, into the cell. Some types of viruses are actually taken inside the cell themselves. Now, you would think that the cell would notice viral DNA or RNA or an entire virus that has been taken in, but in many cases it does not. It takes the genetic material from the virus and it starts following the instructions, which in this case is very bad because the instructions tell it to make copies of the virus. The cell uses its own resources to start building. It starts making so many copies of the virus that it can cause the cell membrane of the host to rupture, explode, lyse. So what happens is now that these new viral copies, they can get out of the cell and go and infect other cells. This is known as the lytic cycle. By the way, the lysing of the cell membrane is a very bad thing for the cell. The cell cannot survive without its cell membrane. The other cycle is called the lysogenic cycle, and I like to remember this is the longer word, so I like to think of this as the longer, sneaky cycle. Viruses that go through this cycle tend to do the same thing at the beginning. They inject their genetic material, but this time, the genetic material stays hidden in the host genetic material. What happens is that when the host makes new cells, it replicates its own genetic material and the viral genetic material. Then those daughter cells make new cells, and they also replicate their own genetic material and the viral genetic material, and so on. This is the lysogenic cycle. That may not seem like a problem, but it really becomes one if the virus gets triggered to go into the lytic cycle from that point. Because then all those cells that have the viral genetic material can start assembling viruses. And as far as what triggers it to go into the lytic cycle, it can vary. It could be a chemical trigger, for example, or a lack of food for the host. Going back to the common cold, a lot of times it has to run its course. Your immune system will typically take care of it, and medications can help with the symptoms so that they don't feel as bad. But unfortunately, some viruses are extremely complicated, like HIV, a virus that can lead to AIDS. Like most viruses, it is very specific. It binds to a CD4 glycoprotein that is found on the surface of specific cells, such as helper T cells. The problem about this particular target is that helper T cells play an important role in protecting the body and the immune system. Since HIV goes after these immune cells, it can make a person that is infected with HIV vulnerable to other infections. There are medications that have been developed to help keep HIV from replicating as quickly, and there continues to be research into looking into ways to keep the virus from attaching, because if the virus can't attach, it can't insert its genetic material. A problem that continues to be a challenge is that many viruses, including HIV, can mutate. So a treatment developed against a certain virus type may not work on a mutated form. So with our talk about viruses, you might wonder, is there anything positive about viruses? Well, some viruses can play a useful role in gene therapy. Definitely something to explore more. Also remember how we mentioned that viruses can go after other organisms, like insects, for example. A virus that targets pest insects could be an alternative to a chemical pesticide. One such example virus that you may want to explore is the nuclear polyhedrosis virus. Still, it's important to consider that whenever talking about any kind of pesticide, chemical or viral, there could be consequences in the ecosystem. Well, that's it for the Muta Sisters. I'll remind you to stay curious.
Alright everyone, so that is chapter 5. Have a great rest of your week, and let me know if you have questions.